Hello, everyone, and welcome to another episode in the Where Are You Now series, in which I sit with professionals and speak to them about their journey. Science isn't just about blood cells, microscopes, and test tubes. Today, we are going to prove that with our guest, Amy Sanders. By day, Amy is an accomplished senior biomedical scientist in haematology, but when the lab coat comes off, she transforms into a powerlifting powerhouse and also dabbles in dance. Amy, thank you very much for joining us today.
Thank you very much for having me here today.
Amy and I have been connected on Twitter or X or whatever you want to call it for many years now. And I think the first time we met was actually at IBMS Congress. And I remember I messaged you on Twitter saying, you can find me because I'm wearing my purple EDTA colored jumper.
Yeah, it was. And I immediately walked into the lecture and I think I was sat about 3 rows behind you. And I was like, there he is. I was also not that hard to spot at the IBMS because either I had rainbow coloured hair or I had my bright pink hair or something. So I was also look for a gap in the crowd because I'm really short and look for the pink hair. So it was always lovely to meet people from the internet in real life.
Yep. I think at the time you had pink coloured hair and you were quite easy to spot in the crowd. I just turned around and there you were.
Yeah, it's a little bit of a beacon. It can be, which is always useful for big networking events.
Yeah. Okay, so one of the reasons I wanted to speak to you was because your journey to become a biomedical scientist wasn't the traditional or the orthodox route that most people take. It's interesting, isn't it? You are now a senior biomedical scientist in haematology, yet when you actually started, you did your BSc or undergraduate in biochemistry. Talk me through that. What was going through your mind at the time when you selected biochemistry?
So I'll preface this by saying I come from a selective all-girls school. So a lot of the time, biomedical science was the fifth option for medics. It was never explained as a career option. I didn't even know there was such a thing as biomedical scientists when I was at school. But I knew I wanted to do something sciencey. I knew I didn't want to be a medic because internal organs should stay inside and away from me. But I knew I wanted to do something to do with like chemistry, biochemistry, things like that, because that excited me. So I applied for a medical biochemistry course at the University of Surrey. Again, was poo-pooed by my school slightly because at that point Surrey wasn't as highly achieving as it is now. But I really wanted to do a professional training year as part of that. So off I go to Surrey to do medical biochemistry. And almost immediately when I get there, people are talking about this biomedical science course. And I'm like, well, that doesn't make sense because Surrey doesn't have a medical school. And then over a couple of years speaking to people on the course, I realised there's this thing called an accredited biomedical science course where you finish your degree and you get to go and work in an NHS lab. And I was like, oh, drat. Why didn't I know about this? But it comes with the environment I was in. So one of the things I've been doing for the last few years is when they have a careers event at my school, I go back and I go, hey, biomedical science, we're a thing, we're a job, and try and explain to them about that so that maybe someone who was in my position now 10 years, 15 years later, God, that makes me feel really old, doesn't do what I did, which is essentially the very long way around. So did medical biochemistry. Turns out I picked 95 of the same modules as the accredited biomedical science course because that was what I was interested in. Just did a professional training year in Finland as part of the Erasmus scheme. So I went and worked in an academic laboratory looking into sleep research because at the time I was like, yep, going to get a master's, going to do a PhD, going to do all these things. Finish that year. No, thank you. Don't want to do a PhD. Don't want to spend 95% of my time asking for money and getting told no. But sometimes knowing what you don't want to do is almost as good as knowing what you do want to do. So it's like, okay, definitely want to be in a lab, but probably want to be in a lab that's doing something, achieving something on a daily basis. Come back, do my master's in health and clinical science at the University of Surrey, which doesn't run anymore. It took over from the old clinical biochemistry masters that they used to run in sort of collaboration with the science, like the STP, the science training program for clinical scientists, because at that point that was what I thought I wanted to do. I wanted to become a clinical scientist. I wanted to become a clinical biochemist. And my undergrad year, so final year, I applied for the STP, got to the shortlist process, didn't get an interview. Master's year, applied for the STP, got longlisted, didn't even get shortlisted this time. And at this point, I'm like, I need to at least find a job. At this point, I am working in coffee shops with a master's, just trying to get out there, just trying to get a job. And I knew someone who I'd met on my master's who had actually done the accredited biomedical science course, had done her registration portfolio at a local hospital and was now working there as a bank biomedical scientist. And she was like, look, we're always interviewing for MLAs. I'll let you know when the next round of jobs is coming up. Like, you're way overqualified, but at least it gets you in. Because by this point, having done the 2 degrees, I knew that number one thing for me was I wanted to be useful. I didn't necessarily want to go into like a big pharma, as they like to call it. Didn't necessarily want to do that. Didn't necessarily want to do research. At one point, I thought I was going to be a cancer researcher. Then during university, came to the conclusion that I loathed and detested microscopes more than life itself, and I was never going to look down a microscope, which, watch this space, Amy. But the hospital was relatively close to me. She knew when the applications were coming up and had spoken to the managers there. I went in and I had a walk around and I met them and spoke to them. So they knew what they were getting into when they offered me the interview, in that didn't necessarily want to be an MLA forever, had the degrees, kind of wanted to be a clinical scientist. So I applied and I get the job, and I started there in 2015, having been in sort of managing coffee shops since the September when I'd finished my master's.
Sure. So before we actually get into your first job working in a lab, just wanted to go back to your undergraduate degree, because you covered a lot of different things, and I think some of them pertinent in the sense that when I was at university, I went through the same phase, same process as you. I went to university, did biomedical science. By the time I had no idea that you could be a biomedical scientist. First of all, that was a huge gap in the careers information that we were provided. I remember we had one afternoon of careers information in which they said you can go into medicine, you can become a teacher, you can go into accounting, you can save the world. But nobody tells you that to do any of those things, you have to either do extra study or get another qualification. Even if it's a biomedical scientist, the thing called the portfolio was not even mentioned. No. We didn't even know what an accredited degree was at the time. Luckily, my degree was accredited, but that was just potluck. I had no idea what it meant or what the value of that would be. But then you said how you did your year in Helsinki. What was it? Helsinki?
Yeah, Helsinki in Finland.
Yeah. And you realized that you didn't want to go into research. And I guess you were lucky in a sense because that was part of your undergrad. So I did a master's after my BSc just to find out the same thing. So I spent a year, $7,500 or so at the time, I think it was, to find out that actually I don't want to go into research. And like you said, sometimes, you do things to find out what you like and what you don't like. And I realized that I don't like research, even though it was interesting, even though it was, stimulating my brain. I just found that I couldn't work in the lab from 9 o'clock. Well, I remember I used to work with zebrafish. So I'd be in the lab from 7 o'clock in the morning to about 7, 9, 10 in the evening. And only to find out sometimes your experiments don't work. All that effort you put in, I was like, you know what, I can't do this.
Yeah, good old zebrafish.
Kudos to people that do it, but, it's not for me.
Yeah.
And that's when I started looking for alternative careers. So you said you worked in a coffee shop in between.
Yes. So
after you graduated,
all during other than my first year, I was really lucky in my first year that I didn't require a part-time job because I had quite a bit I had grants and I also had some help from my family. But from the summer after my first year, all the way through my master's, I was working in coffee shops part-time just for a bit of extra spending money. I did scuba diving at university, and scuba diving kit and trips are not cheap. So just did that. And I mean, I'd worked part-time since I was 16 anyway. So it was just something that was ingrained in me, and that I didn't necessarily, if I could help it, go through all of my student loan. And I know I was very lucky in that situation that I didn't require it, that I did have help from family. But it also meant that I got to meet other people. Guildford is a very interesting town in that you have 2 separate sides. You have the University of Surrey, engineering, academics, science. It's now got its own vet school, which is huge. I think they're probably going to try and get a medical school at some point if I know them. And then on the other side of that, you have the Academy of Contemporary Music, which is an incredibly well-respected institute where you've got some really, really quite famous people who have gone through there. And I was mixing with all that sort of people. And it was quite good because they were all into the same music as me. I did used to get I'd say, oh, I'm really busy with uni, and because I'd have like purple in my hair and a nose ring and a lip ring. Oh, you at ACM? No. You're at Surrey, yeah. What are you doing? Are you doing like dance, music? No. What are you doing? Medical biochemistry? Huh? But yeah, so coffee shops.
Look at you. Yeah. Now, I was going to say, look at you smashing stereotypes,
Yeah.
because not many people associate, like you said, the nose ring, the lip ring, etc., with somebody who works as a scientist. But scientists comes in all different shapes and sizes and colours and appearances, and they're all each as valid as each other.
Yeah, massively. I do quite a bit of outreach with schools and scout groups and things like that. And during the lockdowns, I was doing like Zoom calls and stuff. And one of the days, I just kept my camera off and went, "All right, boys, what does a scientist look like?" And we get the stereotypes of, oh, they're old, they're male, they've got grey hair, they're a bit weird looking. And then I pop up and they're like, oh, like, yeah, this is what a scientist looks like. It's a bit like kind of those, this is what a runner looks like, this is what someone successful looks like we are everywhere, we are not the people you expect, but at the end of the day, like, anyone can be a scientist if that's what they want to do.
Yep, definitely. So then after you had finished your undergrad, you said you did a master's as well. So can you tell us a little bit about your master's and why you chose to do the master's when you knew that you didn't really want to go into research?
So,
because I think you mentioned after your placement year. Yeah.
So I did the master's mostly because I wasn't quite ready to be a grown-up yet. And it had always been in my head that I wanted to do this clinical biochemistry master's because I also knew that I wanted to be a clinical biochemist as far as I was concerned. And at some point I was going to need to do a master's. So my thinking was, I've got the opportunity to do it now while I'm still practiced in writing essays and revising and having that kind of life, and I then don't have to go and somehow carve out time later on, because I'm not great at multitasking with things. I took 5 years to do my specialist portfolio, which I think is some sort of record, but I knew that for me to achieve the best I could, it was better for me when I had the ability to still be at home, stay with my parents, have that sort of support to just crack it out and get it done and just have that tick in the box. Because knowing scientific careers as I do, even if you're not going to go and do research and a PhD, to progress after a certain point, you need that extra bit of paper. So it was take the hit now. Yes, maybe feel like I'm left behind some of my peers and some of my friends because they're already out in the wide world of work earning, but later on I'll have already put in that time. And I am so glad I did, because I admire everybody who works in a laboratory in the NHS doing full shift pattern and everything, who then also manages to do a master's at the same time. Like, I bow down to their powers of multitasking and prioritization.
Mm-hmm. Yeah, definitely. It's not easy work doing a master's while you're also working full time. Nope. I think one of the questions I get asked a lot by students is, so I've done my BSc, should I do a master's? And I think it's a balance between doing a master's for the sake of just doing it because you've got nothing else to do or you don't know what you're doing. And also, you're doing it for a reason.
Yeah.
So I think I fell into the former category. I had no idea what I wanted to do afterwards. And so I was like, you know what? I've got nothing else to do. I'm still in that mode of, studying. Let me just do a master's. It was just something to fill the gap. But in turn, what happened was when I was applying for jobs and I really struggled to get my first, job in healthcare. I'd been working, part-time during my studies, but actually looking for a job in my field, it was a struggle. And then when I actually got my first job, it was because of the research that I'd done in my master's. And so, that really helped. Even though it cost me $7,000, I still, I always think about it. Was it a worthwhile investment? And I think, like you mentioned, having done it at that point in time when I was still in that mode of studying, in that mode of writing essays and doing exams, yes, I think it worked out well because later on in life, I didn't have to worry about, doing the masters on my night shift. I know a colleague who would do all of her studies and lectures and assignments on her night shifts, and that I can't imagine. I struggled enough as it is on a night shift, let alone studying on top of it. So she was fantastic when she did that.
Yeah, massively. And my masters, I was incredibly lucky. So the lecturer who was the course director, I hugely respect him. He's recently retired. I got to go to his leaving talk, and I actually was featured as part of it as someone who can do whatever they want if you put in the hard work, because my second year of uni, I did my exams and I was like barely passing, like 40%, ended up in his office going, help me, and then went on to get a merit in my master's. So he's fantastic. He's an incredible person. Alfred Tumza, he is my god and will always be. But because he'd been very established at Surrey and they'd had this clinical biochemistry course, I ended up getting lectures from some of the eminent people within the field. Gwen Wark, who runs the peptides NEQUAS scheme from Guildford Hospital, she was one of my lecturers. A lot of the duty biochemists that actually worked in BSPS ended up teaching me some of my physiology and things like that. We were having talks from other clinical scientists from Birmingham, from London, from Gosh. So I was exposed to a lot of very successful people in the field I wanted to go in, and that was just because I was really lucky in that I was in Guildford near that hospital, and the scientists there got involved with the course. So when I turned up, it was like, oh, it's you. I remember you from kidney lectures. It's like, yes, hi, I now work here.
It goes to show, doesn't it, if you have a good mentor, how much positive impact that can have on, what comes next.
Absolutely.
From somebody who was getting 40%, like you mentioned, to turn it around to get a merit, that's a fantastic achievement. And, like you said, some of it is down to the mentorship that you had while you were at university.
Yeah, massively. He sort of pointed out where my exam technique could have been improved and things like that. And from there, it was a case of just learn, revise, refine the technique. There's always going to be things that I'm not particularly good at. I mean, if anybody wants to beat my 17% in pharmacology, like at being worse than I am, like I'd be impressed, because I think I probably wrote my name and that was about the only correct thing on that paper. Luckily, as a haematologist, I don't need pharmacology very often.
Exactly. And I guess it also shows that, a mentorship is important, and having a good mentor can be very, very useful. But at the end of the day, it is all about hard work, isn't it? You have to put in the shift, you have to put in the hours, you have to put in the practice to be able to succeed or do well at what you want to do.
Absolutely. At the end of the day, the only person who is going to be able to succeed or fail is you. You can have all the help in the world, you can have all the guidance, but if you're not prepared to put in the graft for it, then it's not going to work. And for me, going on that professional training year, seeing what was out the other side, that was kind of the kick that I needed to just knuckle down, get on with it, maybe stop, like not necessarily like stop going out and that, but pick the times I wanted to socialise, pick the events I wanted to go to, realise that it's really not a good idea to stay up till 2 o'clock in the morning 3 days a week and then be half asleep in your lectures, because you're not going to absorb that information, and when you come back later to those notes, you're not going to know what you were talking about. So kind of upfront sacrifice as such can lead to long-term success. As I've said it to myself several times, just suck it up and get on with it, because it's only for a short time and it's going to pay off.
Yep. You make the hard choices now to have an easier life later.
Massively.
As it goes. So then you started your job as an MLA.
Yep.
And how did that go? How do you find the transition between working in a coffee shop? What skills were you able to bring to the role as working as an MLA? And how did you find that transition between working and studying?
So I was, number one, I was just so excited. Like I was so excited to finally get into a laboratory environment. Yes, all right, I wasn't doing the sciencey day-to-day stuff, and it was a lot of incompletes and posting poo and all sorts of things, but it got me in the environment I wanted to be in. It got me seeing the process end to end, understanding how things work, understanding the process of the tests and things like that, getting to people go, oh, hey, have you seen this? Oh, hey, do you know about this? And for me, even on the bad days, still wasn't a coffee shop, because there's a certain level of arrogance that come with being at the other end of the counter when you're in customer service. I think everybody should work in hospitality and retail because it would make everybody be a heck of a lot nicer. People just think that you're there because you don't have any brains, and actually the people who work in retail, I take my hats off to them because I do not have the patience for people. There is a reason I work in a lab, and it's because I am not the most peopliest of people. I don't have a particularly great filter for crap, so if someone gives it to me, I'm probably going to give it back. So I was very excited to be in a lab, and it was a different environment, especially when I started going on to shifts and night shifts. So that was a real shock to my system, having been someone who is an old lady, and if I'm not asleep by 10 o'clock, like, good luck getting me up before about 7 a.m. But yeah, it was exciting, it was interesting, and for me it was, if I showed the interest and I wanted to learn, people were willing to show me. And I think that's something for people to remember. Like, at the end of the day, it's lovely to learn things in that sort of environment, but you are there for the patient. So people aren't going to go out of their way necessarily, because they do have a responsibility, and you do have turnaround times and requirements for the patient's safety and the hospital demand. But if they have time, if you actively go, hey, I would like to learn this, they will try and find time to you, or certainly we try to find time for people. In that period, I was really lucky because I was still wanting to become a clinical scientist. So I actually got to spend, around my shifts, I'd spend the morning or the afternoon in with the GT biochemists, looking at what they did, looking at the other side of it. Because certainly from a biochemistry point of view, they would be doing the technical validation, but they wouldn't necessarily be doing the clinical authorization, just because of the sheer volume of samples that were going through. And that would come down to the GT biochemists. So it was seeing how they were looking at that sort of thing, the communication they were having with other clinicians, or questions that were coming in and things like that. So that was really interesting for me.
So there's a few things you mentioned that I want to go back to. So firstly, I think it should be a requirement for everyone to work in hospitality for a small amount of time at least, because I think not only does it teach you, how to be a better person, because like you mentioned, people can be extremely vile, disrespectful to people that work in, in shops or in coffee shops or in other hospitality in the industry in general. And I think it will make you a better person, but also it thickens your skin as well. It teaches you about patience. It teaches you about, how you deal with difficult individuals. And I take my hat off to anyone that's done it. The skills you learn there, you probably don't learn in many other places.
Yeah, massively.
And secondly, go on.
It's also the ability to deal with, like, it taught me about, like, people come in for a cup of coffee, they want it quickly. They come in at lunchtime, you're going to have your lunch rush, but everybody still wants their coffee. But you also have to maintain, like, a level of professionalism, and you also have to maintain the standards required by the brand. So it taught me those things, that actually you need them in a lab. You still need to churn out the work even when it's really busy. You still need to be polite to everybody and give everybody the level of professionalism they deserve, even when it's busy and even when things are going wrong. So all of that stuff I learned in retail.
Yep. I think working in the lab, one of the things that we always come across is that when people get stressed out, you notice, changes in their behavior, changes in the way they talk to others, change in the tone. I think, working in the coffee shop like you did, it also, that sense of pressure that comes with different, times of the day. In the lab, it's equivalent to when the GP work comes in, you get that rush of samples, or when the wards are happening, you get that rush of samples at 6 o'clock in the morning. It's maintaining that decorum, that professionalism, that same, personality as you would if it was just, a slow sort of day.
Yeah, absolutely. And why is it that all the work comes in when none of the staff are there? Like, our GP rush used to start at about 6 o'clock at night after everybody else had gone home, and we were on minimum staffing. So yeah, it does set you up, and also it just means that I can't stand people who go out into restaurants and they're rude to the wait staff when it's not their fault. Like, it's different when the level of service is lacking from that individual, but when it's stuff outside of their control, cut them some slack. Like, there's no need to be a complete arse to people. They are human and they are doing their best, and like, this is their job. So like, don't. I did have a moment in the coffee shop where people went, oh, oh, she's got a degree, because someone was talking about the fact that, oh, they'd stayed up late the night before gaming. And someone went, oh, you're going to catch up on sleep. It's like, oh, no, that's a fallacy. There's no such thing as recovery sleep. And I just immediately turn around and I go, I think you'll find there's an increased propensity to stay away from sleep post-sleep deprivation. It is a thing. And they just looked at me and went, that's what my dissertation was in. And it was not knowing that you don't know everything about the people who work out there. Like, they could have degrees, they could have masters, they could have PhDs, they could be a complete expert, and they're just, this is where they've chosen to be for this moment. So don't make the assumption about people. But that was quite funny, because it was about 7 o'clock in the morning and I threw out some big sciencey words, and sort of a stunned silence fell across everybody.
Yeah. I might have to ask you for your dissertation, because I have struggled sleeping. Maybe I can get some tips from that. But in the world that we are today, where cost of living is a serious issue, people have to find second jobs. People, to survive, people work the extra hours, even when they're studying, or on top of the full time, to make sure they can make ends meet. And I think it's extremely rude when you diminish that and you say, why are you working in a coffee shop? You've got a degree. You don't know what else could be happening in that person's life. Some people may just choose to do it because they enjoy it. Everyone's different. Everyone has different things that motivate them. Everyone has different things that they enjoy. And so, live and let live.
Absolutely. And I've always said, like, if I win the lottery, I'm going to open a coffee and record shop, because that's the things I love. I'm a proper coffee snob. I really enjoy listening to music, and I can't think of anything better than having the two together, and having it run how I want, because if people are like ridiculous, I'll just throw them out because it's my place. But yeah, massively, everybody should experience that, because it does make a huge difference to how you treat people going on. I think, like, I've been 15 years removed from retail. I'm still going to give you the benefit of the doubt. I'm still going to be polite, and I'm still going to say thanks.
Yep. So going back to, now bringing it back to science again. So when you were working as an MLA, you mentioned that, you had an interest in becoming a clinical scientist.
Yeah.
So at the time, did you know the difference between a clinical scientist and a biomedical scientist, and what was pulling you towards that clinical route?
So I wanted, and I don't mean to be rude to the biochemists, but they do a lot more. They, in our lab, particularly with the analyzer they had a lot more maintenance, a lot more like kind of tinkering with the analyzers than the hematologists did, and they only did the technical validation. So they were only going, is this feasible to come out of the analyzer? Whereas the hematologists, they also do the clinical authorization, where you're looking at the bigger picture. I know it works differently in some trusts, in some labs. Just with the sheer throughput of work that we had, I mean, now we're up to something like 15,000 samples a day, with 4,500 of those being full blood counts. So it is bonkers. Whereas the clinical scientists were the one, and they were looking at the bigger picture, and they were looking at the changes in abnormal results and whether that was expected from the patient and things like that. And there was a little bit of that with the chemists in the phoning of things, like you were having high creatinines, high ureas, but you knew the patient had CKD. There was different phoning limits for that. But the nitty-gritty digging into the patient history and working out whether this was something that we were concerned about, that came from the clinical biochemist side of things. And at the end of the day, I'm nosy. I like to know things about people. I like to know things about myself. I like to understand the why, and that was kind of where I wanted to be. And again, I didn't have the right degree, so biomedical science wasn't really on my radar. And we didn't have any trainee posts at the time, so there wasn't a huge amount knocking around of like, oh, hey, have you thought about doing this? So clinical biochemistry was the one on my radar, and it was still kind of coming back from school, because that was something that had been mentioned in the careers events at school. Again, biomedical science hadn't been, but a clinical biochemist had been in this weird careers book my school had. So that was what I was still kind of gunning for. Yeah.
So then at what point did you then find out about biomedical science, and how did you end up on that route? And how did you fill in the gap between the fact that you hadn't done an accredited degree and then going on to becoming and registering as a biomedical scientist?
So I applied for the STP for the third time, again, was only longlisted. And at that time, we were then starting to go out to advert for 2 trainee posts for biomedical science. And someone just said, oh, hey, Amy, why don't you apply? And it was a case of I had to really think about it. I was like, right, this is a bit of a pivot away from what it is. But actually, the biomedical scientists, they work as a team. They're all together all the time. Whereas the clinical biochemists, certainly for us, they were off in their own office. There was maybe 1 or 2 of them at best. You were kind of removed a little bit. I was like, you know what, I quite like being part of this team. Okay, I'll apply. Because I can't keep putting my life on hold for a maybe when I've got quite senior people in the lab going, we think you should apply for this, which in my experience means we'd really like you to do this job. Can you go and apply for this job, please? So I applied, and at the same time, I started the process of getting my degrees assessed by the IBMS, which took a huge amount of paperwork and emails and information. So my advice to anybody, if you want to become a biomedical scientist, just please do the correct degree, because there's going to be a huge amount of admin otherwise. I had to get all of my exam results for all the 4 years of 5 years, because I did get marked for my 3rd year in industry. So I had to get all of my exam results. I had to get all of the syllabuses for the modules I sat. I had to get like the course book for each year, which involved, luckily, I was still in contact with Alfred, so I was able to reach out to him, and he was able to get me the zip files with these like course books, because it was kind of before a lot of the digitization occurred. So we were given these things in paper format on the first day of the year. And I didn't have those anymore, but luckily they still existed on the university system. And then I had to go through and justify how each of my modules met the criteria that the IBMS wanted. And it's a very subjective process. I think it is still very subjective. I think it's very much down to who looks at your application as to whether they think that actually it does meet it or not. But I did everything. I included as much information as possible. I said it covered everything as possible. Sent it off to the IBMS and had the interview for the trainee job as I was waiting for the results to come back, because we thought probably going to need 1 or 2 top-up modules, certainly in transfusion and probably a microbiology top-up module, we thought. But again, I'd done 95% of the same modules as the accredited biomedical science course that Surrey run. So I knew that it was going to be very limited, because I compared what I'd done at university to what one of our band 5/6s who had gone to Surrey, who I actually knew quite well. We graduated the same year, and then I decided to swan about doing a master's for an extra year. So we looked at what she'd done and what I'd done and realised where the gaps were and whether what I'd done elsewhere had covered it or not. So I had the interview. I wasn't successful at that round of interviews, but I was kind of shortlisted for future jobs that were going to come up. They said that I was really close, but a couple of other people who had done the interviews a couple more times, had better technique, knew a bit more about what they were doing. I would advise anybody not to do an interview after a night shift, because my brain was not firing on all cylinders. But yeah, so I got shortlisted for that. And then about 2 weeks later, it came back that actually I didn't need to do any top-up modules. And I don't know how I swung that. I have kept that letter forevermore, because I'm like, at some point, someone's going to realise there's been a mistake, and I'm going to say, no, I have this letter. You can't make me go back to uni.
Yeah. So was there a cost involved in when you sent off your degree transcripts and all your module details to the IBMS to review it?
Yeah, I think it was about £150, something like that. And I had to pay that out of pocket to get it done. It's not a huge expense, but still it's £150. But cost that up against the fact that there might have been several £1,000s worth of modules that I may have had to do. I was very lucky that some of the deputy general managers really believed in me, and they were already looking out for partial funding and scholarships from various universities to get me through these modules, because they were really keen to have me sort of stay and be part of the team. So to have that buy-in from very senior management was again another reason why I decided to apply for the job, because if they want me that bad that they're trying to find money for me and I'm not having to find it myself, like, I need to give this a go.
Sure. So you said your first experience at the interview after a night shift, it wasn't great and you weren't successful.
Yeah.
So then when did you actually become successful in getting your trainee post?
So I was lucky in that I was told that so there is a process within the NHS. If you interview for a role and you're put in the talent pool, if another role comes up within 6 months, you do not have to re-interview for that job. I have been the beneficiary of such a policy twice now. So I was told that I would be next to get a trainee post. So the two people got from the interviews that I had got their jobs, and I was told I would be next. Because there were a couple of people who were trainees and they were just taking a while. But in the meantime, I was just continuing my work as an MLA. I was learning as many things as possible. I was becoming as useful as possible. By the time I finished my job as an MLA, I knew how to do every single bench except one, which was haematology senderways. But I knew how to do chemistry senderways, which was actually the harder of the two, I think, because there is so many more tests. I have to go so many more places.
Yep.
Whereas 95% of haematology goes to the Royal Marsden, as far as I'm concerned. But during that period of waiting, our biochemistry lab manager left and he went to a different network. And then he emailed me a couple of months later, "Hey, how you doing? What's going on? Do you bioscience yet?" "No, I'm still waiting." "I've got some jobs going up. Why don't you come down and come on a tour here? By the way, it is in haematology." Okay. And it was actually at my local hospital. I was very lucky my commute to work was only about 35 minutes, but the commute to the local hospital would have been 15. So I was like, "Oh, I'll go down," because they've offered it to me. And I had a walk around and I had a chat with people. And then they were like, "Yeah, apply for the job, see what happens." And I applied. And then suddenly this invite to interview comes up. And I had a bit of a crisis of conscience. And I ended up going to speak to my lab manager and went, "Just so you know, this is going on." And I didn't have to do that, but the lab I'm in, they've been very good to me. They've given me a lot of opportunities. They've let me go to courses. I've asked to do things and they've said yes. So I thought I'd give them a heads up. And I went, "And by the way, you did say I was next. Why am I hearing words that there's someone before me?" "Oh, well, it's a haematology job. We didn't think you wanted haematology. You say you hate microscopes." At this point, I was like, well, this job they're offering me is going to be in haematology. So it's the same thing. I'd rather stay here. All right, the job's yours. Now, that could have been seen as one of two ways. I'm leveraging the fact that I'm leaving over there, but I was promised the next job was mine. So wherever I would have gone, it would have been a haematology job. And I don't know if I'd have ever got the job at the other hospital, but at least they were offering me interview based on seeing this very new, very scared person and going on what my previous manager had said about me. So I ended up getting my trainee post in October of 2016. I did not learn a single thing while I was a trainee biomedical scientist because we didn't have enough MLAs. And I was just still on the MLA rota, still doing all of those shifts. But every so often someone would grab me and be like, "Hey, look at this down the microscope. Hey, look at this result. Hey, look at this." So there was the occasional thing. I came off the MLA rota 2 weeks before my portfolio verification. So I learnt nothing as a trainee. I am very pleased to say that for the most part, that has changed. And we are now getting people in as trainees properly, early, teaching them things, getting them up to speed to the extent that I didn't realise someone wasn't state registered and I almost let them start validating on their own without a babysitter until she went, I don't have a registration number, I've not done my portfolio. I was like, "Oh, well, you're that good. I didn't know this. So that's on me."
Sure.
But yeah, I did all of that and then I state registered, having done my registration portfolio in July of 2017.
Okay. Before we get on to your registration and your actual experience as a trainee, one of the things that I wanted to pick up on, and it was quite a surprise to me because I've personally not worked in the NHS apart from a short stint at Colindale, which is NHSPT, for 3 months after I graduated. All of my jobs after that, whether it was as an MLA or a trainee or then doing my specialist, was in a private pathology network. And so I've not worked in the NHS per se properly. So when you mentioned the fact about if you do an interview for a particular role, that's valid for 6 months, you said?
I don't know whether this is NHS wide or whether this is our trust, but the trust I work in,
Right.
if you interview for a role and you are kind of reserved, you don't get the job, but we reserve you because you're quite good and we would like you to work with us, but we don't have as many jobs for you. You do not have to re-interview for that role if one becomes available within 6 months. This
Sure.
has paid off for becoming a trainee, and it also paid off when I became a senior.
Sure. That makes sense. I guess that's something people need to investigate themselves to see whether it is something their
Yeah,
local laboratory offers or
yeah,
not.
because
Okay.
you have to
So,
think about the process as well of putting out all these job adverts in the NHS. So you've got to rate, you've got to check with all the finance that you've got, like the funding, and you've got the role available. You then need to put the advert out on track. It's got to be up there for X number of weeks. You've then got to collate all your answers, shortlist people, like long list people, shortlist people, invite them to interview, do the interviews, offer out the job, go through all of that. Like, if you can take someone who's already passed all of those requirements, you've just saved yourself 6 weeks, 8 weeks of all of the admin in the back of house, and get them in earlier at the end of the day.
yep, of course, that makes sense. So, moving on to your actual experience as a trainee.
Yeah.
I think most trainees can attest to the fact that as a trainee, you're just a glorified MLA, because we do a lot of the things that are required for the portfolio anyway. And that's one of the advice that I give to people who are wanting to become a trainee biomedical scientist and going on to becoming a biomedical scientist, is that when you're doing or when you're working day to day as a MLA, you can look at the portfolio, it's available online, and you can start thinking about which evidence you're going to collect while you're working as an MLA in haematology, in specimen reception, in transfusion, in micro, in any of the disciplines. You do everything that is required for the portfolio, and you can start thinking about what evidence you're going to collect, how you're going to present it. I know one of my previous colleagues, he started making comics because that was, he was very good at drawing and he enjoyed making the art, and he made comics of some of the activities that he did. For example, health and safety, how he'd clean up a spill. So he drew a comic, and that he then, when it came to him actually writing his evidence for his portfolio, when he actually got a trainee biomedical scientist post, it was easier because he'd already done it. He was like, there you go, I just color it in and it's all done. And so it saved him time that way, and he managed to finish his training very, very quickly because he'd already done most of the graft beforehand.
Yeah, massively. And we're really lucky in our lab that quite a lot of the MLAs we take on have got the degree in biomedical science, and they do want to become biomedical scientists. So some of them have got their portfolios paid for by our trust, like by our network. Obviously, this is on a network by network basis, so the rules will vary. And we're happy to sort of pay for the portfolio, but we do sort of explain that, look, there's not a guaranteed job at the end of this. Again, we have a couple of apprentices who are doing the same route, going through higher qualifications, going through university. For those guys, we will try and find a job for them because obviously it's a 4 to 6 year long game. So we've invested quite a lot more money in them, quite a lot more time and energy. But when you're looking at the long-term benefits of that, like you've got that familiarity with the trust, with the way things work. But a lot of the MLAs, like even if they're not doing it, I'm like, hey, just go pull up the reference copy, have a look at that, see what you're doing, see if you can build stuff. We have two very active training officers who offer a huge amount of encouragement, who are happy to do the marking, look at things, arrange interviews. So there is quite a formal process within our trust about how people go through and progress on these portfolios. And everyone gets involved with like helping practice on the tours and things like that. And certainly when I was like doing stuff for my specialist, was like, okay, so ask me the hardest question you can think of, because if I can answer this question, I'm not going to get flummoxed on the day. But yeah, with the training portfolio, I did a lot of my stuff on night shifts. Night shifts in MLA are certainly can be less hectic than they are as a BMS. So certainly on the weekends, I was drawing up evidences, but because I was trained in a lot of areas, I was getting quite a high variety of evidences, because I was getting things from when I'd been in transfusion, when I'd been in sendaways, when I'd been doing incompletes, when I'd been on the phones. So it was good to get the range of evidences, and also the verifiers, they like to see different kinds of evidence. They don't just want you to do like a witness statement. They don't just want you to do a reflection. They're going to like things like process diagrams and spider diagrams and pictures and annotations and stuff like that. So the more variety you can get in, the happier they're going to be.
Definitely. I think one of the things that you mentioned previously, quite some time ago, was about how, as biomedical scientists, being nerds, we like to talk about what we do. And so when an MLA or somebody comes up to us, telling us, asking us, can you show me this? Most biomedical scientists are more than happy to do it. And remember, I used to be on night shifts, and like you mentioned, sometimes nights there's a dip in the busyness, and so it gets a bit quiet. I didn't want to say the Q word, but now I've said it.
Oh, you've done it now.
And I've done it now. I know the curse of the Q word. And so there'd be moments during the night shift when there wouldn't be much going on. And the MLAs would come up to me and be like, hey, can you show me something under the microscope? What are you looking at? Or how do I do a blood group? And I would enjoy teaching them how to set up controls in BT, like not actually doing patient samples, but, using expired cards, because we should always have expired cards lying about. So getting them to set up the controls and seeing how to interpret the blood groups. Also showing them things on a microscope. We had a microscope with a multi-header, so you could fit 3 people on it. So 1 person driving, 2 people observing, and so we could sit down and just look at things together, just point out abnormalities or what a normal cell looks like, what an abnormal cell looks like, and just get that discussion going. And they really enjoyed it. And I think it also comes from being curious, having confidence in your curiosity. There were those MLAs who would ask you questions all the time. How do I do this? Or what are you doing now? Why are you holding this, FPC back when the hemoglobin is normal, but there's something else? They ask you because they're curious, and so you teach them. And there's others who may not be that, forward with their questions, but they're still very interested. And so I would, if there was a quiet moment, I always find that teaching happens best when you're actually doing something rather than sitting down just talking, passively. So if they were, doing something on the analyzer, I'd be like, why are you doing that? And what does that tell you about, how the FPC works, for example, or how the analyzer functions? And it gets them to think about what they're doing too. The fantastic MLAs are fantastic at what they do, but for them to understand what's happening in the background, which I don't think always gets portrayed to them, is another skill as well. So, why do you flush the flow cells? What's the point of it? What does a flow cell do? How does it work? That's something, we cover at a specialist level. But, just as general knowledge, I would enjoy passing it on to the others around me. They would have an understanding of what they're doing, and hence they can then do it better.
Yeah, massively. And it's like, at the end of the day, a laboratory wouldn't work as efficiently as it does without an MLA. Yes, it needs state registered biomedical scientists or HCPC registered biomedical scientists because we are the only one that release the results. But the MLAs are vital parts of that machine because they are doing the stuff that facilitates what we do. Like I can't look at a film if someone hasn't loaded it on an analyzer to make it. I can't report a malaria unless someone's run the kit for me. Yes, I could do those things myself, but there are so many other requirements of my time that they are there and they help massively. And at the end of the day, if I ever needed help on a night shift, I'd be like, hey, can you come with me a second? Like massive hemorrhage and your printer stopped working, you're having to handwrite everything. Hey, I'm going to write this out. Can you double check the details for me? You teach them, they get on with it. They're fantastic. They're incredible people. And I don't think they get enough credit for what they do.
Totally agree with that. Yeah. Emily's like the gel of the lab. Without them, the whole thing would stop running, stop functioning. I mean, as a biomedical scientist, sometimes the things that we do so infrequently that if we were asked to do it or we had to do it, it would take us so long to do that, I'm glad that Emily was, looked after that responsibility. It would take me twice as long and I'd probably get stuck at least 4 times, not know what to press on the screen.
I mean, our haematology senderways, one of the things that they are responsible for is staining bone marrows. I wrote that SOP. I have done it once in my entire life and I did it wrong. Luckily, I didn't put any bone marrows in before I decided I'd done it wrong. But I've written the SOP, but because it's something I've never done in my life and we were so short staffed, it was like, can you just do this? Can you read through the SOP and do it? Whereas they just bash it out like it's no problem. And there's things I check with them. I'm like, how many microliters is this? Yes, I could go and look in an SOP, but they can pull that information off the top of their heads. Whereas for me, I might do that test once every 3 weeks, once every 4 weeks, when I'm like, it's quicker if I do this, just give me this information, please. So they are fantastic. But I do agree with you, there are people who have a more active curiosity and those who are more passive in the way they learn. But that goes for everybody. Like you'll find some people are dying to learn to do something new when they become HCPC registered, and there's certain things that they're really excited to learn, and there's some people that it's like pulling teeth. And somehow
Yeah.
microscopy is the one that is like pulling teeth for some people.
Yep. Okay, so moving on to now, you have finished your portfolio. First of all, how long did it take you to finish? And then when did you start doing your specialist? Did you go straight onto the specialist, or was there a gap in between where you worked as a band 5 biomedical scientist?
So I became a trainee in October 2016. I did my state registration in July 2017. I think if you look in the front of my registration portfolio, it took me a year. It took me 12 months, and during that 12 months, I was on full shift pattern, lots of night shifts, lots of late shifts, all the weekends known to man. I yeah, I was in the lab a lot. But that was mean I was getting exposure to a lot of things. So I became registered in July 2017. I think I went straight into blood transfusion because we were quite short of members of staff. So I'd actually learned quite a bit more than the normal MLA would in BT before I'd done my registration portfolio. Just because we were very short of BMSs, I was seen as someone who was going that way anyway. So anything I could learn as an MLA, they taught me. So I learned how to maintain the analyzers. I was looking at what stuff needed panels. No, I wasn't releasing it because I couldn't, but I was going, oh, based on that pattern, it probably, and the fact that they're an antenatal patient, that probably needs a 4-cell. Based on that pattern, that needs a full one. And they were teaching me stuff like that because they knew what was coming later on. With my biochemistry background, we all thought it'd be a great idea if I did the brand new shiny blood sciences portfolio. So I applied for that and I got that in the January of 2019. And then sort of sat on it for about 6 months. But I started doing it. Started working my way through it. At the same time, I'm also trying to become competent in everything. No, hang on. 2018, I got it. I was state registered in 2017, and I got it in 2018. And I started working through the portfolio and started doing everything to do with that. And I'd finished the haematology stuff. And then because I had a master's and I was working on my portfolio, again, this is specific to our trust, our network only. It's going to be different everywhere else. But I was told that I could essentially sit a viva and I could do a written exam to the level of the specialist portfolio. And if I passed it with I think it's an 80% pass mark, our network sets, I would then be elevated to a band 6 on the provision that I finished my portfolio. Because my master's was in a related subject. It was health and clinical science. And I was working on the specialist portfolio. So I did do that in December of 2018. And I passed that. But I'd put that off for probably about 6 months after I'd gone fully on shift because I was being a wetbag. And I was like, I'm going to fail this. I'm not good enough kind of thing. Very much doubting my ability, despite the fact that I was signed off on morphology, signed off on HPLC, which not everybody in our lab was. That was and is still a bench that we are fiercely protective over because it's for the sickle cell and thalassemia screening program, and you cannot get that wrong. So we are very picky about who goes anywhere near that bench. You tend to only touch it if you're a band 6. So the fact that I was working to that band 6 level, again, meant that that was one of the reasons I was allowed to go near that bench. So yeah, and then became a band 6 in 2018, 2019. I can't remember. It kind of blurs at this point. And was working at that specialist level as I'm still slowly chunking my way through the specialist portfolio and getting some time with the chemists to start doing the biochemistry section.
Right. So you were working as an MLA in biochemistry, right? And so all of your
it was blood. So the MLAs are very much blood sciences. They will do haematology, biochemistry, and transfusion. They will rotate across all of the roles there. Whereas the biomedical scientists are still split currently, even though the plan was to make it all blood sciences. It's still very much haematology, transfusion, biochemistry. But the thinking was, because I had the biochemistry background, the biochemistry degree, but I was a haematologist, that was why it was suggested that I did this blood sciences portfolio.
Sure. So when I applied, or when I spoke to my manager about doing the specialist, I asked for them for the blood sciences portfolio as well. The reason being, when we did our registration portfolio, it was
Yeah,
rotational. So you would do special reception, you do haematology, transfusion, and biochemistry. So we'd rotate between the four specialisms as part of our registration portfolio. So I said, you know what, I'd like to do biochemistry as well. But at that time, like you mentioned, we weren't set up to have that rotational side of things. It was hematology and BT or
yeah,
biochemistry. And I chose heme and BT. I preferred it. But one of the questions I wanted to ask you was, you started off as somebody who enjoyed biochemistry and ended up in hematology. But what about all the other specialisms? There's genetics, there's micro and viro, there's cell path, histopathology, there's so many
yeah.
others. So do you ever feel, did you enjoy them at university? Or was the choice to do biochemistry based on something else? It was the only one that appealed to you at the time.
It's probably 50-50. So cell path and microbiology, I didn't really enjoy them. Like, I enjoyed them in the sense of the learning, but it wasn't something that I could see myself doing in, day out. Looking at the biochemical pathways and stuff like that, that always interested me more. And then from that, sort of the hematology, how it feeds into the, like, how things work, that was always more of an interest to me. Genetics and I do not get on. I came out of my cancer genetics exam crying for reference. That was how badly that went. So, and with me wanting to become a clinical biochemist and things like that, and then the fact that the hematology lab was in the same place as the biochemistry lab, it was kind of the natural kind of lead in. If I was going to do anything, it was going to be blood sciences.
Sure. So you're working as a band 6
Yep.
now, and you're still working through your portfolio,
Yep.
your specialist portfolio. What was the transition between, having just registered and becoming a band 6? And did you find it difficult to go from being somebody who would rely on others as such, to get things second checked, or to get, because you're still quite new at that point. When you've just registered, you're still quite new, you're still
Yeah.
learning. And then when you're a band 6, it's almost like, okay, go on, do
Yeah.
it.
So a lot of it was kind of get dropped in and hope for the best. So the lab I work in is incredibly busy. It's one of the hub sites. So we do a huge amount of work. My argument is we probably do 2 years worth of work per year versus everybody else. Looking at the figures on a daily basis, we are the busiest lab in our network by 40%. And that's versus another hub. Our quietest site
Wow.
will sometimes do about 350 full blood counts a day, and we're at 4,500. So the sheer exposure you get to all of the weird and the wonderful and the strange stuff, it happens quite quickly. Add on to that I am Twitter's favourite magnet for all of the bad things.
I was going to come on to that, yeah.
I have a reputation that seems to precede me along the lines of, "Oh, Amy, not again." I think it was only my third night shift in transfusion, I get a call. Patients just whipped out this letter from their immunologist saying that they're IgA deficient and that they'll go into anaphylactic shock. We've just spiked the bag of blood. Can we give it to them? What? No?
What?
Huh?
Yeah.
Shoved, and I've not heard of this. Shove my head out the door, get the other person on the shift. IgA deficiency. Yes. I can't give them normal blood. No. Don't do that. Which leads to me Googling quite sufficiently overnight, with me going, please don't give that to that patient. They're going to go into anaphylactic shock on us. Speak to
Yep.
NHSBT, speak to the haematology consultants. And it's just things like that. You come across the weird and the wonderful at 3 o'clock in the morning, and then you learn from it. And then you're the only person in the lab when everybody else is on a meeting or going home, and you've suddenly got what we were on Siemens 2120s, and we had the beautiful triangle of doom of that's a semicircle of neutrophils and eosinophils with no platelets. Oh my god, I think I've just found
Yep.
APML. There's nobody to look at it down the
Yep.
microscope for me. Technically I am signed off, but I would like a supervisory adult. Nope, guess I'm my own supervisory adult. Look at it. Myelocytes, promyelocytes. Oh god, call a haematology consultant. They come down, take one look. Yeah, it's APML. Well done. whip it off, go and get the patient. So it's things like that. It's just, sometimes you're just going to get dumped in it, and sometimes you're going to get a chance to learn about things, and someone's going to be there to talk you through the process and why we're doing what we're doing. And sometimes you just have to learn on the spot. And as I taught people when I was teaching sort of massive hemorrhages and things like that, you only have to be 2 steps in front of the clinicians for them to think like you know what you're doing. And sometimes you're learning as they learn, but if you are calm and collected and ask them for all the information you need and you seem like you're in control, you can be that beautiful swan that we talk about, where underneath it's frantically going, oh my God, oh my God, oh my God. But you will learn, and then you will remember, and you will either never do that again or you will teach other people about it. But that was certainly my trial by fire. But because I was doing so many night shifts and so many out of hours shifts, I think that was why my learning curve was so steep. And also, I'm nosy. I want to learn. I want to know. I want to know why that's doing that. And as I was doing the specialist and I was learning things, certainly the blood sciences one, there is nowhere near as much haematology in there. So for context, the blood sciences portfolio, Section 7.1 of the haematology portfolio is the entirety of haematology for blood sciences. So you don't do any special coag. There's nothing on malignancy, nothing on hemoglobinopathies, anything like that. So it was very much a brief overview. But again, that's what you want in a generalist. So you don't need all the nitty gritty. But I was getting the nitty gritty just because I'm me, and I found one acute leukemia, a TTP, and a CML in the same week.
Oh, wow.
Like I said, magnet.
Yep. And I guess, you mentioned that working on the night shifts or working out of hours, there's always less staff. And sometimes you may be the only person trained in that department. But having said that, there's always, it's always okay to ask for help. It's always okay to get a second opinion, whether you're in transfusion, in chemistry, in any of your specialisms. It's always okay to ask for help, and there's always help available. Sometimes it can just be calling the reg on a call, calling NHSPT. NHSPT have some fantastic consultants, and even the people in RCI are fantastic. When you call them and you ask them for advice, they give you fantastic advice at all times. They're always available 24/7. I know I've had so many issues. Transfusion was always my weaker point. I was always even though I enjoyed it more at the beginning, hematology I used to struggle with a little bit, but transfusion, it was just there was just too much to it, and I just couldn't get it in my head. And I would always struggle with, do I do this or not? Because it's also scary, because transfusion has an almost immediate effect on the patient. Within hours, you can see the effect. If you make a mistake, it's all it takes a couple of hours, and so that would always, scare me a little bit too. But generally, there was a lot of things I didn't know in transfusion. And so I would be on the phone to NHSBT and, fantastic colleagues over there who explain everything, who would help you out whenever you need. So that was a lifeline for me.
Yeah, hugely.
And even, your other colleagues around you, having a second pair of eyes. Sometimes you see something on the microscope, and like you said, you know it is probably this, but you want somebody else just to confirm it for you. And if you're the only hematologist working in the lab on that day, the only other person might be the hem reg. And I guess it also depends on how your hospital and the lab is, located, because sometimes you have labs which are on site, and so the hematologist can just walk up the stairs and the reg can just come to the lab and have a quick look. But sometimes they're actually off-site. So I've worked in a hospital lab where the lab was 15 minutes walk from the actual hospital. And so for the doctor to come along, it's a lot more effort and a lot more difficult. And, sometimes they'd be like, just take a picture on WhatsApp and send it to me.
Yeah, we were really lucky. We had Celavision. So for those people who don't know, Celavision is a semi-automated slide reader. It's not AI, it's partial AI in that it'll have a bash at doing a differential for you. But it is very much dependent on how good the quality of the film is, how old the cells are, which bit of the film it's looked at. But Celavision certainly can be a lifesaver, because at 3 o'clock in the morning, when you've got nobody on site, because we didn't have 24 cover by the consultants, they were on on-call basis out of hours. So at 3 o'clock in the morning, when you find an acute leukemia, you can go, hey, it's on Celavision, can you have a look? And then they can make the decision of, is it a 3 o'clock in the morning problem, or can this wait till 6 a.m.? But again, you also then start learning as you get more and more experienced. If I wake them up at 3 versus if I wake them up at 6, they're not going to do anything till 6. So you know what? Let's just get them in a better mood at 6:30 in the morning and not wake people up. But when you've got that one of, is it, isn't it? Oh, I don't like this. And you can get them, they can look at it and go, well, I can see patient history. Whereas in the lab, a lot of the time we can't see anything. We're running off of clinical details. And
Yep.
if someone would put routine in clinical details one more time, I swear. But we're running off of clinical details and what we know about the patient, whereas they've got everything there that they can see because they've got all of the clinical systems. So they can then make a way more informed choice than we can.
Yep. I think with improvements in technology whereby electronic health record systems are now also encapsulate the limbs as well, allows the people in the lab to see the patient history. That's quite useful. But when you're using, standalone limb systems, you've got a different system, which is the limbs. You've got a different system, which the clinicians use. You've got a different system, which the pharmacists may use, which the antenatal midwives may use. And so it's very hard to get the communication between them. And like you said, the number of times we've had full stops in the lab as, clinical details or unwell. What the hell is the patient doing if they're not unwell? Why else are they not?
Doctor's request. That's a lovely one as well.
Yeah, or tomorrow's bloods. So why am I getting it now? There's all of these, and if we got paid £1 for every time that happened, I think the lab would be rolling in money. We'd be doing fantastic research, but alas, that's not the way it goes, is it?
No, it's not. And I don't as well, unless you speak to people and go, this is all I can see, right? So if I could have some information, case in point, we had one of those, I don't know what this is, I don't like this, I don't like this. Two of us looked at it and went, that kind of looks blast-like. And it was Boxing Day night at 11:30. And I just go, you know what, I'm just going to call them. Because it's an out-of-area patient. And I have a discussion with them, look, anything else going on? We go through it. Oh, and they are also currently undergoing treatment for AML. Oh my God, that'll be why it looks like a blast, because it kind of is. Oh, can you please put that in your clinical details in future so we don't have a heart attack down here? So it's things like that. It's because now our hospital is a bit better in that we can get in and see some of the patient records, but it doesn't seem to be standard who can see what. I can see a hell of a lot more than everybody else can. I don't know why, I don't know how, but I can. But it means I can go in and I can see the notes and things like that, and I can look at their medications and I can work out things like whether they're anticoagulated or not when clinical details on the limb system say they're not, but they've got a APTT of 150. Things like that. I can go in and I can have a look before we make the decision, because a lot of times you then get the sort of pushback. Why are you calling me about this? Of course they're on XYZ, but I can't see that. I don't know that. So if anyone clinical is listening to this, please tell your labs their clinical details that are helpful, because we will love you forever.
Yeah, I mean, sometimes there are some doctors or some other clinical staff who provide you beautiful details. And I remember once, one of the doctors wrote such beautiful details, I melted in my seat at how gorgeous it was. Angels started singing in the background when I read it. It was so good because it explained everything that was happening with the patient, what drugs they were on, what their previous history was. And so rather than me having to chase things up and call them and say, oh man, oh shit, I've got this really abnormal result, I could just say, oh, this is known, don't worry about it, it's all fine, I just released it straight away. So it turned a problem which would have taken me 15 minutes. Sometimes it's very hard to get hold of clinical colleagues. They're busy too. Yeah. They may be in clinics, they may be on ward rounds, and so they may not have access to a phone, so it can be really hard. But when you've got nice clinical details, you can just say, oh, this is expected. I don't need to inform them. I just put a comment in, they can see it when they need to see it.
It makes all the difference.
It makes it so much easier. Yeah, definitely. So talking about your trial by fire, working on the night shifts and while doing your specialist portfolio at the same time and working as a band 6. So at what point did you then decide to apply for a senior post?
So it would have been the end of 2020. So we had one of our band 7s was moving on to somewhere else. And we were, because the notice period for band 7s is 3 months, we petitioned to sort of HR and payroll to, can we get the ball rolling a bit early? Yes, we know he's going to be in post, but if we have to get someone in from externally, it's going to be months and months and months, and we can't afford to not have a band 7 for 6 months. Couple of months, fine, but we need to have someone like coming into this post. And I was one of the more experienced band 6s, simply because of, not necessarily by years, but certainly by exposure. By this point, I've been state registered for just over 2 years, 2.5 years. So I'm still a baby. If anyone ever looks at a band 7 job description, it goes 5 years post-registration experience. I was nowhere near that. But my argument is I've done basically 2 years of work a year. So why not? Why not me? And if nothing else, it would have been the experience. So I applied for the job, got offered to interview, and the closer it got to the interview, the more I realised, actually, no, I think I could do this. I think I could do a really good job because I'm already being used to train newbies. I'm already doing a lot of the health and safety. I'm already being involved in the UCAS process because people know I'm going to do it properly. I'm already doing a lot of the more advanced techniques. I'm doing microscopy, I'm doing HPLC, I'm in BT quite a bit. Why not me? So again, with that beautiful thing of Twitter, I reach out, and I was very lucky to get quite considerable coaching from Kip Heath, who is a lovely, fantastic human being, and she helped me through the process, like gave me ideas about what I need to talk about in interview. The ever-present Danny Gaskin as well gave me some assistance and some advice on the interview process. And there was quite a few people interviewing. There was a few of us internally, and there were some external candidates as well. And I go in, and I've got the least experience. And I go in, and I try my hardest. I answer all the questions. I try and use the big buzzwords, and I come out of it. And normally you find out on the day or the morning of, or they let you know. And my interview had been quite early in the morning and hadn't been on a night shift, so I could actually string a sentence together. And I tried really hard, and they went, "Oh, we'll let you know." And apparently I set the cat amongst the pigeons because they weren't expecting me to interview or be as prepared as I was. So I caused a little bit of a furore and a very difficult situation, which I like. I like that it wasn't clear cut. And essentially what it came down to was I hadn't got as much experience as one of the other people. But there we go, that 6 months in the reserve pool kicks in. They knew, and I didn't at the time, that our laboratory manager was leaving. So they had to go and speak to one of our very, very experienced band 7s and go, would you be up for applying for his job? Because Amy's done really well in this interview. We don't know what to do. Yes, all right, she'd be up for applying for the job. So they knew within that 6-month period it was going to be a knock-on. He would leave, she would apply for his job, and then I could drop into her post and we wouldn't need to interview. And that's exactly what happened. I didn't get the job in the January, but I did get offered the job in the April when everything had moved along. And then COVID happened, just so become a senior. So yeah, that was how I became a senior. It was again a very convoluted thing, but I am always a case of, a lot of my career has been a case of, want to bet? Why not? Why can't I do this thing? I like to say that I am 95% motivated by spite, and the other 5% is an overriding requirement for caffeine. So that's how I became a band 7. 6 weeks after I became a band 7, we had a UCAS audit. So that was a trial by fire, because I not only had I become a band 7, I'd also been dropped in as the quality officer at the same time. So I was having to learn all of that process and all of that background and the requirements we needed for UCAS. And the world is on fire. So we're on a day by day. Everything is changing. Processes in the laboratory, processes in the hospital, staffing in the laboratory, the way we're staffing the laboratory. Are we going to have 2 people on the night shift?
Before we get on to
everything.
Before we get on to the whole COVID part of the story, I want to go back to, the beginning of you applying for your senior post. You mentioned some fantastic names, Kip Heath and both Danny Gaskin. They're both fantastic biomedical scientists, and they're both very, very approachable and helpful. And I'm glad to know them as well. Like you mentioned, sometimes you have to ask yourself the question, why can't I do it? And even if there is a gap between what you currently do and what you're going to be doing, you can always bring that up at the interview, and you can say, you know what, I'm willing to grow into the post. And I find people appreciate that. And the other thing that you mentioned about, setting off a cat amongst the pigeons because you were less experienced compared to the other applicants, it just goes to show that sometimes it's not about the time that you spend in a role. People can spend years and years and years and just coast by and do very little. And then there are others who may only spend a fraction of that time and achieve so much more within it. And I had the same sort of a similar scenario when I applied for my training portfolio many years ago now. It seems like a lifetime ago. So I hadn't actually worked in blood sciences. So at the hospital that I worked, coagulation was a separate department to blood sciences. So coag, all of routine coag and specialist coag was done separately, and you'd have hematology, transfusion, and chemistry. They were separate. And I was an MLA in coagulation. I'd never worked in hematology or transfusion or chemistry. But when I went for the interview, I was able to demonstrate my willingness to learn, my willingness to adapt, my willingness to take on new information. And unfortunately, that upset, and when I was successful for that training post, it upset a lot of other MLAs in the lab in blood sciences because they'd been there for donkey's years. And they were like, we've been here for, 5 years or 6 years or 7 years. It's not fair that other people from other departments can come in. And I think, sure, there is that aspect, but I also think sometimes it's about how you portray yourself on the day and how you show yourself, to your other staff around you. I wasn't actually going to apply for that trainee post. Reason being, a few weeks ago, I'd had another interview at another hospital, which I totally flopped. It was so bad. I was still suffering from the trauma of that. And I was like, I'm not ready for another trainee post interview. But then one of the seniors in my lab said, the manager for hematology has asked, have you applied for it? If not, you should. And like you mentioned, when people higher up say you should do something, there's probably a reason why they're asking you to do it. And so I was like, okay, fine, I'll apply. And I did my revision the night, at the time leading up to the interview. But having not worked in biochemistry or hematology or transfusion at the time, it was a struggle because, I'm somebody that learns from doing things. And having not been in those departments, it was quite difficult for me. But, I crammed in as much of the knowledge I could from books and the internet. And when I went for the interview, at the end, they asked me, is there anything else, we should know about you? I think I've told this story many times. I spoke about honey. I had a cold at the time, like I do now. I seem to have colds all the time. And I said, you know what? People have been telling me to take honey and lemon. And I wanted to see if there was any scientific basis for that. So the type of person that I am, I logged into PubMed. I actually looked to see if honey has any, effect or if it's one of those tales that people tell just for the sake of it. Their mom told them, and so they're telling, other people. But turns out honey does have antimicrobial effects. And so we spent more time talking about honey than we did about biochemistry, hematology, transfusion, or quality. And in the end, I didn't say much in the hematology bit or the chemistry bit or the transfusion bit. I answered the questions, but not as great as I would do now. Now that I've got some knowledge and some experience of working in departments, I think I could have answered them so much better. But the next day when the manager spoke to me, she said to me, you blew us away with your willingness to learn and your knowledge outside what you need to know. And that is a quality that we're looking for. And so it's the honey that got me the job rather than my knowledge of, the actual departments, which is quite surprising. But like you mentioned, your story has been a trial of fire throughout, and that's been a running theme.
Yeah.
So when you became a senior, did you find there was a big jump between working as a Band 6 biomedical scientist and then taking on the responsibility of a senior? Because as a senior, you are the lab manager in the lab. The lab manager is outside the lab doing all the other duties, but you're managing the lab within it. And so you take charge of rotors, like you mentioned, quality, health and safety. Did you find it was a big gap or a big jump, sorry? Or was it a case of you've done a lot of this before and so it was just leading on and building up to that?
So I kind of like to say that before I became a Band 7, I was a 6.5. I was doing the 6 work, but where I could, I was taking stuff off people's plates. So I was doing the health and safety. I learned how to become an auditor. I was helping with training and stuff like that. So I'd kind of dipped my toe into that pool as such. So I thought I knew what was coming. Becoming a Band 7 is a whole different bag. Number one, the interview is different to anything you'll ever do because by that stage, we expect you to know the science. So the entire interview, no one asked me about science, but a couple of very brief questions. It's not like a 5-6 interview where we're going to grill you and be like, these are the results, what you're going to do. It was very much about process and quality and that sort of thing. But it was the juggling that came with being a Band 7. In an ideal world, you're going to get a couple of days off bench. You're not necessarily going to be pulled into the rotated number of staff that are required to man the laboratory, so you can get on with doing all your SOPs and your training and the competencies and stuff like that. But that never happens because someone's sick, someone needs annual leave, someone's short, you're having to double up on staff because you're training there is always a lot of requirement on your time to do things, and you've got quality department asking you for things, you've got other departments asking you for things, you're now that person that everybody's name knows because you're the senior of the lab, so everybody asks you the question, so you've got consultants coming to you about things, and I'm sure people don't mean it, but when there is a senior in the same room as you, people like to divest themselves of their brain and just send all decision making in your direction. There's a responsible person here, there's a senior here, they can make the choice for me. You know they can make that decision because they do it at 3 o'clock in the morning when you're not there. So it was also learning how to nicely phrase, what would you do if I wasn't here? Because I'm happy to teach people and let people make decisions and give people ownership of what they do. That is the kind of manager that I am. I'm not going to sit here and declare that you must all do it this way. I'd rather you come up with the process and get to the way that you want to do things yourself. I might offer some pointers if I think you're absolutely going to crash and burn, but how are you going to learn if you don't try? And I'm here to take the flak if it all goes a bit wrong, within reason. But people liked to outsource their brain to me as a senior, and I'm there looking at the films, answering 4 emails at once and trying to get some sort of competency or training matrix or something updated. So there was a lot of juggling that came with it. And it's May 2020, so there's a lot of external pressure and stress and worry. What am I bringing home?
Yep. I think it also comes back to responsibility as well, isn't it? When you're working on a night shift and you're on your own, it's different to when you're surrounded by other members of staff who you can give that responsibility to. I think people sometimes are scared of taking responsibility for the actions and the decisions that they make. And so say, there's a senior over there, ask a senior instead of me because I don't want to, I don't want to be put down as a person who's made this decision. Like you mentioned, the big topic of the time was COVID.
Yeah.
And you've just become a senior. And things were changing. I remember, guidelines were changing on a daily basis. And there was always that uncertainty because we didn't know what it was at the time. How do you cope with the uncertainty? And how did you continue to do what you were doing without, freaking out, to put it politely?
Yeah, there was a lot of freaking out. So my partner worked in hospitality. I think, again, why I'm so adamant that people should do it, because he has been there. He did the eat out to help out, and oh my God, they all deserved like a medal for that. So I was terrified that I was going to bring something home to him. He's already had pneumonia once and he doesn't recover well from it. So I was terrified that I was going to bring something home. I was going to give something to a friend or a loved one. And a lot of the time, it wasn't even that things were changing daily. You'd turn up at work, it was one thing. You'd get to lunchtime, there was something else. By the time you finished, it was something else. And by the time you came back in the next day, something had changed again. So that I don't deal well in a constant state of flux. I do suffer quite badly with anxiety. So that was at an all-time high. Very, very high, very, very stressful. But at the same time, it was just kind of get your head down and get through it. So we were trying our best to keep the guys in our lab safe. So we then, normally our late shift was at minimum manning, but we actually split all of the staff so that more people were on the late and the core shift, because the GP shut as well. So 90% of our workload just vanished overnight. So we were only dealing with the truly urgent inpatient stuff. And again, we were seeing massive shifts in what was happening with that. Never seen so many D-dimers in my life, and sequential D-dimers as that, like same patient 4, 5, 6 days in a row. We just don't get that normally. But once that turned out that it was quite a good prognostic tool, off we went running with that because it was already in the lab. So for the first couple of months, it was running out of D-dimer reagent left, right and centre, because we've never done this many in our lives, and we've gone from needing 2 boxes in the fridge to get us through to suddenly we need 4 boxes at all times on both analyzers. So it was scary, and not being able to see family and things, and it got quite lonely, because it was literally go to work, come home, go to work, come home. And I have like a really strong group of friends from various things. My best friend, I've known her since I was 5 years old, so she's stuck with me for life. So it was staying in contact with her, making sure she was safe, making sure my other friends were safe, and trying to arrange Zoom calls and all of that for people. But one big thing for me that went was the gym went. And suddenly the one thing that I used to keep my mental health in check was gone. So if I never see a burpee again, it will be too soon. Because
Yeah.
rather than going and lifting heavy weights, I live in a flat as well, so I don't have a huge amount of outside space. I am lucky that I do have a courtyard, and that was the year with the glorious summer. So I was outside in that
Yep.
courtyard doing various CrossFit workouts with kettlebells that I managed to buy 2 days before the lockdowns were announced and things like that. So it was a very strange
Sure.
time. Everything was different. It was the new normal. Like if someone had gone, here, have a deadlift bar, I'd have probably cried, because it was just what that one tiny piece of normality would have been something for me. But yeah, it was weird.
Yep.
And if I think about it too much, it's just kind of like, how did we do that? How did we manage that? But we did, because
Yep.
what else could you do?
Yep. There was no other choice,
No.
was there? And I think it's a testament to your flexibility and your growth as a individual and as a scientist that despite all the challenges that were thrown at you, there's a lot of uncertainty and you handled them well enough. And here you are today. It's a thing of the past. COVID, it's still around, but it's not as serious as it was when we first experienced it because of the unknown. We knew very little compared to what we know now. And, you've done fantastically and you are here today.
Yeah.
And I remember one of the things that happened to me was I was working the late shift at Guy's Hospital, and Guy's in St Thomas's is a HCID unit. It's a high consequence infectious diseases unit, and so one of the first people with COVID came to St Thomas's, and I was working the late shift in transfusion. No idea what's happening in the world around me. Doing my sickle cross matches to send out for the next morning. And my sister she sent me a message. She's saying, "Oh, by the way, there's a patient with COVID at your hospital. Don't come home tonight," because it was on BBC News. I was busy working. I hadn't had a chance to check the news yet, and so she was like, "Don't come home." And it was just it was meant as a joke, but it just goes to show the fear of the
Yeah.
unknown at that point. When people were dropping like flies in certain places, it was very, very scary. And I remember, when the guidelines changed from how the sample swabs were being collected, first of all, they'd be packaged up in 3 times, sent to us in a special box. And then they said, "Oh, it doesn't matter anymore," within a space of hours. And we were like, really? There was a lot of uncertainty and a lot of unknown. And I can only, sing praises of all the people that were in charge, all the seniors, all the lab managers, who probably had so many sleepless nights, making sure that the lab was running safely, and not just the lab, across healthcare, across all other professions, making sure that their staff were safe. And sometimes they would put their personal comfort to the side to make sure that their staff were, comfortable and they had all that they needed for, doing their job in a safe manner.
Yeah, absolutely. And I mean, for us as well, like there would be times where there was a couple of times where my partner would wake up and he'd have, because he was in contact with the public, he'd have like sore throats or stuff. The minute that happened, I would literally be throwing myself out the door. And we're all masked at this point in the lab. We were masked until May 2023. It was a very, very late that we unmasked. But it would be like, right, get out, get COVID testing, don't come back, let us know what's going on. And he actually got pinged Christmas Day of 20, it could have been 2020, yeah, Christmas Day of 2020. He got pinged, you need to do a 14-day isolation. You've been in the vicinity of someone. It was when they were doing like all that stuff on the phones. And I then had that.
Yeah.
He's been told to isolate. Do I have to isolate? What's going on? I'm supposed to be on a night shift tomorrow. What's happening? So things like that. It was even sort of after that first big hit of everything happening, it was still the onslaught of, still the uncertainty for 12 months, 18 months afterwards, just with it all ticking along in the
Yeah.
background. But yeah, like you said, stuff would come down like triple bagged or double bagged and be opened in fume cabinets with isolated centrifuges, and nothing was allowed to be
Yeah.
outside of anything, and all this, even after staining, the films were put in methanol and all of this business. And then suddenly it just went, and it was like, no,
Yeah.
it's normal. don't worry about that. And it was just, okay, so is it because it isn't as bad as we thought it was, or is it because we've just gone, it's everywhere, we can't stop it? And I never got
Yeah.
the answer to that.
Yeah, but I guess we're here today, so that
Yeah.
is, whatever they did, some of it must have worked, and some of it must have been, good guidance. Moving away from, the scientific side of things, I wanted to touch a bit more on, you mentioned your mental health during that period, and some of the things that you do as a person to maintain your mental health. I know you're a powerlifter. How did you get into that? And what do you think? Why do you do it? I guess my question is.
So I suffer with something called trichotillomania. My eyebrows are a lie. These are actually microbladed onto my face because when I am stressed and when I am anxious, I pull my eyebrows out. Some people do it with their hair, some people do it with their eyelashes, some people do all three. I just do my eyebrows, which is probably the least bad of the three. But it can get to the extent where I look like an alien and I have nothing left on my face, and that is massively crippling to my confidence because you don't realize how much an eyebrow makes a face until there aren't there anymore. And just the stress of going out and knowing that I'm probably going to have to draw them back in halfway through the day when they rub off and things like that. So that has been something that I have suffered with on and off since I was 16. We have peaks and we have troughs. At the moment, I'm at like a new baseline where it's not great, but it's not as bad as it has been. And also because of the microblading, I don't notice it as much, so therefore I don't get in that feedback loop. For me, powerlifting and movement in general is hugely beneficial because it gives me an hour, 2 hours where I can shut my brain off. I'm waiting to be assessed for ADHD because we think that might be something to do with it. I can have 4 trains of thought at once going in 4 different directions, and I ain't got a clue what's going on. And just being in my head, it is incredibly loud in here. So powerlifting, because all you've got to do is just stand it up. But when it's 100 kilos on your back, that can be the only thought in your head of just get up. So it gives me a time where it's quiet in my head. Powerlifting I found at university. I got to my final year of my undergrad and went, I don't recognise this person I've seen in the mirror. Like, I have put on weight. I am not the person I remember being. I was very active growing up. I'm actually a national level ice skater and ice dancer. So you can see the very clear progression to powerlifting. It totally makes sense to go from sparkles and glitter to unitards and chalk and talcum powder. But I started doing some like weight training in the gym with just some machines and some dumbbells and stuff, and progressed from there, and then started doing a little bit of barbell work. And I mean, when I started, I couldn't bench the bar. I still can't bench my body weight, but I can almost. But I couldn't even bench a 20-kilo bar. And then someone introduced me to a deadlift, and it was love at first sight. I lifted it and it felt awesome, and I was like, this is really cool to be strong and to be able to do this. So it progressed from there with more and more resistance training. And then one of my friends up in Scotland, she was a power lifter, and I was in sort of a non-commercial gym environment where there was powerlifting and there were people who competed and stuff, and they did a little local just in gym meet, and I was like, oh, I'll do this. And I did, and I very much enjoy working towards a goal. I'm very goal orientated, so I'm always better when I have like an end date to work towards. And it was also very nice because it wasn't subjective in the sense that skating could be, did they like your dress? Did they like your music? Did they like your style of skating? Where were you skating in the group? Was it after somebody really good? Were you first on, so they had to use you to set the median mark? That sort of thing. Whereas in powerlifting, it's, did you pick it up? Did you follow the rules? Were you the strongest on the day? So that for me is very nice because it's very clear cut. And it's just sort of gone from there. I'm never going to lift the most. I'm never going to be the strongest. I'm never going to set any records. But when you're 4'11" and you can deadlift 140 kilos, it's pretty awesome to say that to people because they go, huh? How much?
That's a huge amount. Yeah, definitely. And when you were talking about your condition earlier, you mentioned about your eyebrows and picking at your eyebrows. I think the eyebrows are one of the parts of your face that nobody normally thinks about unless they're not there. You can shave off your beard, you can shave off your head, and it's fine. It's normal. But when somebody shaves off their eyebrows, or gets rid of them, or they fall out for whatever reason, it makes a huge difference to your
Huge
facial
difference.
appearance. And so I can only imagine how much impact it must have on your self-confidence when they're not there.
Yeah, it is. And I remember when so I've been through therapy twice now for them. When it gets really bad, it's like I need to go back, I need to get better coping mechanisms, I need to kind of address what's going on. I'm a perfectionist with very poor boundaries. It's the long and the short of it, who refuses to ask for help, and my support system is kept at arm's length because I don't want to bother anybody, is what's come out of 2 rounds of therapy. I'm getting better at it. I'm learning to let things go. I'm learning to relax. I'm learning to give myself some downtime. But the day I went to the GP, I walked in to the GP room and I went, I need some help with this, pointed to my face and burst into tears because, yeah, it might not seem like a big thing, but when you are so conscious of it and it makes a huge difference to what your face looks like, yeah, you're going to have some massive body confidence issues, and it can be debilitating. Like, even on night shifts, I remember taking everything with me. So at 3 o'clock in the morning, if I'd rubbed my face, I could go and draw my eyebrows back in. And that should be the least of my worries at 3 o'clock in the morning on a night shift. But it was something I was so conscious of that I finally decided that, you know what, I'm going to use some savings and I'm going to do this for me. Because it was always a case of, oh, you'll just get over it. Just get over it. You don't need to do it. And it's like, well, why do I need to get over it? Why can't I do this for me? Yes, all right, work on getting better, but why not make this quick fix now that's going to have a huge impact on me? And it has. It's been life-changing to have had them microbladed in, because it just means I don't have to think about it in the morning anymore. It's not something that I say it's a blessing and a curse. My mental health was clearly displayed on my face for all to see. How's Amy doing? And it's there on my face. Whereas now I kind of have that privacy with how's Amy doing. Well, she has to tell us. Her face isn't going to tell us for her.
Yep. I think a lot of people take that for granted. When you ask people how you're doing, you ask them because you can't see it.
Yeah.
But like you mentioned, it must have been quite a challenge when it's, like they say, it's written over your face.
Written all over my face.
Yeah. And on that note, we've been talking for more than an hour and a half. And on that note of self-care and taking care of yourself and your mental health, I'd like to end this episode. Amy, thank you very much for joining us and sharing with us your experience.
No problem. It's been a pleasure to talk to you. And yeah, I know it's not been the normal way round. I know it's been the long way round, but it's definitely been worth it.
Yeah. It's been a fantastic journey. Thank you for taking us on a ride on your journey and showing us how you got to where you are today.
No problem. Thank you very much for having me.